There you go. Okay, so we will record all the sessions so that you guys can go back and access this later. And then you can also access the speaker files so that you can use those to show your students if you want to, give them a quick intro to any of the things that we learn about. So welcome, everybody. My name is Megan Heafy. I am the director of the Rural Teacher Training Initiative. Until recently, Kristen Boskin was the director and I was the project coordinator. So I am new to this role. I am also still doing my old role. So a little busy, but I'm here to support you guys. So whatever you need, feel free to reach out, and we'll go through a little bit today about how I can help you guys. So let me get the PowerPoint started. All right, can we see?
Yes.
I wish it didn't make you guys small. Okay, I'm gonna go back to my big grid. I'll figure it out. That's oh, there's the drag. Okay. All right, that's more of you. All right. So welcome. And so this is Biotech Infusion. And I work for the Georgia Bioed Institute, which is part of Georgia Bio. So we'll go over a little bit about what that is and what we do. First off, so again, I'm Megan Heafy, and I'll let Jeff introduce himself. Jeff is going to be our co-host today.
Hello, I'm Jeff Rapp. I'm director of the UGA Bioexpression and Fermentation Facility as of August 1st.
Yeah, so previously Jeff was at Athens Tech directing their biosciences program. So he's a great resource to learn about the technical college system and then to learn a little bit about some things at UGA that you may not have heard of. So, and they have a great tour at the Bioexpression and Fermentation Facility. So hopefully we'll get some of you guys hooked up with that. All right, and so welcome everybody. So you can see on the map, there are people from every corner of the state participating. So welcome, welcome, welcome. So if you guys could, in the chat, you can just pop your name, number of years that you've been teaching, where you're teaching, and then whatever subjects you're teaching, or maybe you're going to be moving into something next year that you want to share so you can start gaining resources. Feel free to put all that in there. I'm going to try to find my chat window. Oh, it moved up there. Perfect. Yeah, welcome. All right, and I'll put that in the chat so that when I move on, you guys have it. Awesome. Oh, yeah, we have some first-year teachers, some veterans, some people looking toward retirement. There's what we're putting in the chat, just so you guys have it. And a little bit about my background. I taught high school science for 9 years in the metro Atlanta area. So I did an alternative preparation program in Clayton County to get certified. My bachelor's was in biology. And they said, hey, you can teach chemistry. So I got certified while I was teaching. And then I went to Gwinnett County and taught at Duluth High School for about 6 years. Teaching chemistry, obviously, biology, and then I got AP and gifted certified, so teaching the AP bio and gifted. Did a little bit of forensics, so I kind of stayed away from physical sciences, but I'm here to help you guys see how you can incorporate biotech into those courses because there are plenty of overlaps. Awesome. Thanks, everybody. And probably for the next meeting, we'll have you guys pop your names in the chat again, just so that Jeff can help take attendance and make sure that we count everybody and give you credit for attending. Changing Chromebooks. All right. Yeah, so some technical difficulties out there, but that's okay. The recording will be available if you guys get cut off. So if I could bother Mark and Trisha real quick to introduce themselves. So Mark and Trisha are doing some split screen there so that you guys can see their materials as they're working today. So we have Mark and Trisha Peterson from Paulding County High School.
Hi, I'm Trisha Peterson. This is my 19th year teaching, and I've taught everything from physical science, co-taught biology, honors biology, co-taught environmental science. And now I'm teaching biotechnology, and I'm teaching AP environmental science and AP biology.
Thanks, Trisha. Is Mark, there you are.
And I'm Mark Peterson, and I've been teaching for 19 years as well. And I've taught a little bit of everything. Most recently, I'm teaching AP Physics, AP Chemistry, and Biotechnology Independent Research and Honors Chemistry.
Thanks, Mark. So you guys can see that they cover pretty much everything. So if you have a question, don't be afraid to ask. So in your binder, everybody should have one. Their information is also in here, along with, excuse me, Cynthia Greer, who will be with us at the spring training. So hopefully there are paper copies of pretty much everything. I've been marking on my screen. I didn't know. Okay, so a little bit about our organization. So Georgia Bio is the parent organization, and that is basically the life science affiliate for any company in the state. We also work with universities, basically on advocating for life sciences, whether it's lobbying, helping them with relocating staff, and really anything that they need. I help them to get rid of things that they no longer need. So if they have expired consumables or old glassware, old equipment, I can take those items and then we allow you guys to come and pick them up from the equipment depot that I'll talk more about. And I see that Maria is on. So Maria, if you want to just pop on and say hey real quick.
Sure. Hey, everybody. It's great to see a lot of familiar faces from the community. Good to see you, Jeff. And of course, Megan, thanks for pulling this together, especially the transition from Kristen. It's very exciting for us to have so many teachers kicking off our 5th year of this initiative. And I'll also do a shout out to our industry speaker, Eric at Persone. And of course, the Petersons, wonderful to have you guys on. I'm Maria Thacker. I'm President and CEO for Georgia Bio. As Megan already said, we're the trade association for the industry in the state of Georgia. We are a 33-year-old organization that has worked to drive forth the business community in the life sciences. We have biotechnology, pharmaceutical, medical device, diagnostic companies, and of course, academic institutions as part of our membership. We really do a lot of work in advocacy, economic development. I work closely with the state on companies that are expanding and moving to Georgia. And we just connect the community and have a big network. I will say that the Institute has been a personal passion of mine since I started over 17 years ago. I was a second full-time staff, and I'm very grateful to help have gotten our original workforce programming off the ground and been able to see where Megan and past team members have taken the Institute. So we're thrilled to have y'all participating. I know you have a lot to consume, and listening to me is not fun, but and I'm home with my 6-year-old for fall break, so I will not be staying on because I think I've only distracted her for about 5 seconds. So wonderful to see you all. I really cannot thank you all enough for everything you do, not just participating in this, but everything you do for your communities and for the students you teach. I know it's not easy. I fully acknowledge the work that teachers have and the job you have, and I commend you all for everything you do every single day. So thank you, and thank you for considering bringing the life sciences into your classroom. With that, I'll turn it back to you, Megan.
Thanks, Maria.
Thanks, Maria.
All right. So I probably won't spend a lot of time on this because we're going to cut into the teacher or our speaker's time. But you guys are participating in the Rural Teacher Training Initiative, which we have 5 components. So you guys will be placed in cohorts today. Just half of you will go with Mark and half will go with Trisha. You guys will learn hands-on biotechnology and how to incorporate that into your classrooms. You will work with your master instructors. We will provide equipment and supplies, and so you will get those to implement labs with your students, and then we'll introduce the equipment depot at a later date. So hopefully, you know, I've experienced professional development before, and we really have tried to make this something that is valuable for you guys. So If there's something you want to see that we're not doing, just let me know. All right. And I do see that Eric is here.
Hey, Megan.
So we can go ahead and we'll transition to our speaker. So I will stop sharing. All right. And thank you so much, Eric, for joining us today. So I'd like to introduce Eric Doherty of Persone, and I will let Eric tell you more about himself.
All right. Hey, thanks, Megan. Everybody that's on, really appreciate it. I know it's been a long day already for you, and I do appreciate the time and hopefully the attention as well. Again, I'm Eric Doherty. I'm president of Persone. If you haven't heard about us, you will. We are a kind of a three-pronged attack company. I've got a diagnostic division, I've got a IT division, and then I have actually a global component. We can talk through kind of all three of those. My background's all in healthcare. My dad was a pharmacist on the Walgreens side, so I'm a kind of a Walgreens brat as far as growing up is concerned. Probably unique to you all from a student standpoint. I went through, I think, 14 different schools between kindergarten and high school, being a Walgreens brat, kind of an Army brat, moving all over the Midwest and the South. And in Johns Creek, I got two boys that are over at UGA, go Dogs, and definitely really a huge proponent of education and tapping into that. I am a mentor over at UGA, an executive leadership mentor, and I really feel it's a passion of mine to give back to the community in that regard. So just wanted to make sure that that was passed along. Thank you very much, Michelle. I agree. And I do have a son that graduated from Mizzou as well. So there is a little bit of competition in the next couple of weeks. So we'll see how that one goes. So let me kind of talk through kind of what I do. And then, anybody who has questions, please feel free to, raise your hand and I'll go ahead and answer those. And again, open forum. So please don't hesitate to ask. I've got some slides I'll go through, but just to kind of walk through what we've done. I started the company 3 years ago. Through COVID, as you kind of imagine, back in 2020, we had originally licensed out technology out of the University of Florida, out of Gainesville, and it was new tech. Basically, it was what's called electrochemical biosensor technology. So I'm going to do a raise of hands. How many of you have ever heard of electrochemical biosensor technology? You can go in the area for reactions and raise your hand. I'd love to see how many people really know what that is. I don't see anybody yet. Okay. So in essence, it's similar to glucose strips. If you remember the diabetic patients who might have a handheld device and you've got a strip, basically you've got a handheld unit and you would have a strip design. And so on that strip design, depends on how you structure it. But with our technology, it was electrochemical base. And so you would use gold as a surface conductor and then basically apply a solution or liquid to that strip, put it into the device, and the device obviously would have a charge, like a battery charge or electrical charge. Push the button, you get a readout, you get a wave voltammetry readout. And so what we found was that we'd get a positive or negative out of the test, depending upon what you were testing for, but then also get a data composition out of that testing information because of the wave voltammetry. So we started the pathway down kind of two directions, where you were working on the diagnostic to bring that to market and then working on the IT framework. Well, lo and behold, about 6 months into that, found out that the technology from University of Florida was not commercially viable. So really had to make a pretty big pivot. And as the leader of the company, basically pushed back all of the IP and patents back to University of Florida. Sorry, Gator fans. And then basically developed our own IP and own internal kind of documentation around our technology. And then in the midst of that, we had the IT side of it ongoing. So we ended up developing a relationship with SAS, which is the data analytics company out of North Carolina. And so through that, then we developed that partnership where they would basically take the data and then run with it and obviously do analytics out of it. And so the pets are, oh, sorry about that. Just thought it was for me. And then basically the data would be, again, analyzed and obviously provided back to practitioners. Well, as that started building, we ended up asking SAS, what would they like to do from a project standpoint globally? And so that's kind of where we're at today, where we're actually right now in the midst of a AI generative, obviously, revolution, as you all, I'm sure, have heard over the news over the last couple of months. But in essence, what we'll be doing is running data on a grand scale for AI generative analytics. So that's kind of the background on the company. So let me go ahead and dive into some slides and give you some heads up. So one of the things I think is really beneficial for us is, what we've looked at from a disease state standpoint. Going back to the healthcare side, our focus right now is on sepsis. And if you don't know, sepsis is a killer. I mean, you've heard about cancer, you've heard about heart disease and diabetes, et cetera, but it's amazing how many people are affected by sepsis or with sepsis, but also how many more people die from it. And so, we hear about the COVID numbers, obviously, over the last couple of years were obviously very, very high. But when it comes to sepsis, 11 million deaths globally each year, that's a statistic that we obviously want to minimize. And so that's our goal with our system going forward. And what we've done is this platform here. And again, it's on the IT side, but it does cross over into the diagnostic side and kind of the electronic health record side as well. So follow me through a healthcare continuum for a patient. And I'm sure a lot of you either have been through this type of scenario, maybe you have family members that have been through this scenario, or unfortunately, maybe have kids that have gone through this scenario, where you've got a child that goes into a hospital setting, they go into an emergency room or emergency department, they're seen by a doctor, they're then moved into maybe ICU or critical care to obviously have more testing done. And then they're starting to feel a little better. They move into a hospital room, and then they're either discharged at that point in time or maybe a couple of days later discharged, but they end up going over to the left-hand side where you have the skilled nursing facilities and long-term care facilities, or they go to the right side, which is the home or the hospital at home, which is kind of the new age of remote monitoring for home patients. And in essence, what we're doing on our end is basically collecting any and all data points within that continuum. So think of anything from an electronic health record system, lab information system, lab tests that are being done at the bedside, any type of IoT, any type of wearable data, any type of monitor, anything that has a data push, we're connecting into our system, being at the hospital, at that rehab or nursing home facility, or at the home environment. And what we're doing is cleaning up all of that data. So in essence, as you can probably imagine, garbage in, garbage out from a data standpoint. The benefit, obviously, of good data is having that good solid front on a data composition standpoint. So what we're doing on our end is cleaning up that data, and then we send it over to SAS. And what's really great with their system, and again, if you're not familiar with them, they're one of the largest data analytics system companies in the world. They do pretty much most of the clinical data analysis for pharmaceutical companies. They do all of the clinical data analysis for FDA. If you've ever gotten a fraud alert, probably SAS in the background from your credit card company. They do a lot of fraud work with banking institutions, but more importantly, from a safety standpoint, they do all the avionic data analysis on your jet engines around the world, as well as manufacturing. So they are a huge, huge organization out of Cary, North Carolina. And so basically what we'll be doing with them is throwing data to them, and they'll be running the information through basically multiple algorithms of information at the same time to provide real world analytics on that information. So keep in mind, let's say you're looking at a sepsis patient, is what we're looking at primarily at the beginning. But if you move over to a cardiac patient, ischemic heart disease, a diabetes patient in the endocrinology space, COPD, or even asthma, or even kidney disease, these are our 2024 kind of guidelines for moving forward. But as you get that data into the system, it's obviously going to be a very, very large amount. And so we'll be actually providing information back to the practitioners to show how that patient is doing throughout that whole world of ecosystem from a healthcare data standpoint. So I'm going to stop there, see if anybody has any questions first. Okay. And then basically on a granular scale, this is more in scope of what we're doing going forward within both the hospitals, but also the research institutions. This is really showing, again, how our system is set up. We're very agnostic from a data standpoint. We look at anybody or everybody as a data provider. We don't care what electronic health record system someone would have, or what lab test you have, or what device you've got as far as a monitor goes. We'll be pulling all that data in and then moving it into either the hospital setting or the research setting. So from our standpoint, again, the aspect of taking the data, basically enhancing and seeing how much better it can be, and or from a treatment standpoint, making sure, you know, patients can be treated better with better medications or better protocols. Ultimately, that's what we're trying to drive towards. So it's a pretty big task. It's something that, you know, most of the health institutions that we're already connected into, like Johns Hopkins, Emory here in Atlanta, UGA over in Athens, St. Louis University, Advent Health, and Wake Med. There's a lot of folks that need something like this where they can find real-time analytics, provide better information back to the practitioner to help the patient in their healthcare journey. So a huge undertaking, but a lot of fun building it up. Any questions? Nothing yet? Okay. Let me go to the other part of our company then too. So one of the other things that we're doing kind of lies within what you all do from a teaching standpoint every day is basically our Global Connect Persona Connect project. And what we're doing, and we're working potentially with UGA on this, we are already tapped into University of Central Florida, that in essence, those lower to middle income countries around the world that need what we have in the United States and in Europe, as far as better medications, better way of treating, better diagnostics, et cetera. We're looking to provide that globally over the next 10 to 15 years, which obviously is a huge undertaking, but a huge benefit to the global marketplace. And really that's been allowed with technology. You know, on the left-hand side, and this is actually a pictorial that was done back in the 1920s, where again, this was how the first radio picture for a TV was set up years ago. And on the right-hand side is Elon Musk's company and the ability to actually tap into satellites from a technology standpoint allows us now to actually provide that global healthcare benefit. And that's what we're striving towards within our system. You know, there is obviously a global need. There's the global infrastructure in place, which is great. The opportunities there, and really the solution is there too. We've got products, we've got therapies, we've got diagnostics that can actually make a difference globally. And so from our standpoint, it's a huge win-win for not only, you know, a company, not only for the healthcare organizations that we'd be partnering up with, but also ultimately for those patients. And we've got a big driver for us. We've got Dr. Redfield on my team, the former CDC director. If you remember, he also did a heck of a lot of work back in the day under George Bush #2, you know, helping folks over in Africa with HIV and AIDS. So he's still well connected over in Africa and obviously around the world with a lot of the health ministers. So he's joined us to really push this effort forward in making sure that we can help people around the world on a number of different disease states. And our aspect of it is really kind of looking at training folks who are in those lower to middle income countries on the ability to be using diagnostic tests and diagnostic tools, therapies, et cetera. So once they're trained, they can then go see patients out in the field. You know, there's a lot of NGOs, there's a lot of faith-based organizations that are out there that are helping and trying to help people around the world. This is another kind of aspect of tying in the work that they're doing currently, and then obviously expounding on that. And so we'll be basically moving this forward in June of 2024 with the help of obviously organizations like UGA and UCF, and as well others, to really train folks both from just a naivety level healthcare basis all the way up to nurses, and giving them the ability to actually, you know, use products, test and treat people in the field, and really make a difference from a health standpoint globally. So huge benefit. And really the aspect of all of these different disease states around the world. I mean, malaria is not a big one here in the United States, but globally it's a massive problem. And so there are easy solutions to what we have built. And so we're definitely going to be rolling this out in the next 6 to 8 months. All right, questions. Let me see here. No questions. Fascinating by pads. Oh, I'm sorry. Yes. What does SaaS stand for? So SaaS, it's not the software as a solution, not the SaaS. SaaS is the organization, it's a company. So you can go to www.saas.com And they are a huge data analytics company. They're very agnostic, so they don't care where their data comes from as well. So they would just run the data analytics off of the information. But they are a massive company from an analytics standpoint. Cool. Thank you for that. What training is needed? Okay, one of the questions is what training is needed for these healthcare workers on a global scale? So within the University of Central Florida system, they have a virtual training program in place. They've got 16 languages. We want to boost it up to about 53 or so, which is kind of the Facebook level of languages. And so from that, they would be basically, they've got a tiered section of training. So you've got basically the basic training. So how do you use a thermometer, pulse ox, blood pressure cuff, that type of thing. And then moving up from there, other diagnostic tools, other diagnostic tests, et cetera. So their training is level centered to where, depending upon the practitioner, are they a naive healthcare worker? Are they a nurse healthcare worker? Are they a potential doctor healthcare worker? And through those different tiers, they have different training levels. Hopefully that answered your question. Other questions? Okay, back to you guys. My question to you guys. How many of your kids are thinking about AI? You guys can go off mute, I think. Megan, can you take them off mute?
They can, I think they can take themselves off mute.
Okay.
So yeah, y'all can shout out now.
Yeah.
100%. Yeah, this is Michelle Sizemore. I have a lot of teachers who are thinking about it, but whenever I've talked to students, they're not thinking about it as much as the teachers are, but a lot of the teachers are thinking about it.
That is interesting. I thought the kids would be on the front edge of that and start really kind of thinking through what that might do to them from an implication standpoint. I don't know if you guys saw or on LinkedIn, JP Morgan's head, Jamie Dimon, came out and said that AI will actually reduce the workday level to about 3.5 days a week. And then on top of that, the employment numbers will also be obviously scaled very highly back. So that was that came out today. So
I know there's a lot of controversy about AI and the use in schools. It's great for corporations, but at university level, there's pros and cons that I've heard just stories on the radio with professors talking at, at Harvard and other stories I've heard. So I think there's still a lot of concerns about it as well.
Well, and again, the reason I was asking that question too, having two boys at UGA, y'all ever heard of ChatGPT? Because your kids have. Yes. Every one of your students has, I'll guarantee it. And so good and bad, but that is kind of the unfortunate wave of the future. It's if you haven't used it, be careful, but it is fun. If you do it right, it's amazing what it can do, from anything from a recipe to a full-on white paper. I mean, it really can provide pretty stellar background information that is hard to decipher it from what could be typed out by a human.
My students aren't so interested in AI, but they are interested in healthcare. So I find this very interesting for them, because a lot of them, are concerned about trying to get that 4-year nursing degree and things like that. But this seems like kind of a in between there for them. So I find that very interesting.
Yeah, I think what we're finding on our end is with the CAP system, the ability to, provide more. I'm going to call it provocative data, because it's not being done today. Good example, you've got a patient who goes into a Piedmont hospital here in Atlanta, and then they're transferred over, or they go back into a Emory hospital, and then they go into a Grady hospital. None of them talk. So your health records that happen at Emory aren't shared at Piedmont, that aren't shared at Grady. that's a dilemma, especially if you are a cancer patient or a cardiac patient, et cetera. So the ability to tap into your own data and have the ability to, provide that across a network and networks is kind of what we're looking at doing. Advent Health, I'm sorry, not Advent, Mayo and Cleveland Clinic are very siloed organizations where they try to keep the data in-house. And so we're trying to break that, I call it monotony of data, because you're not helping the patient. You're helping the institution, but you're not helping the patient. And going back to the nursing side, I think where the benefit lies, and let me kind of show you what we do on a screen standpoint, so to kind of give you a heads up. So this is a critical care dashboard. So whenever you would go into a ICU or critical care hospital room, basically this would be what would be showing either on the screen in the room or at the nurse's station. And so what we're doing on our end is the ability to trace and track data through basically these trend analyses. So going back to that sepsis patient, if you're monitoring their heart rate, you're monitoring their blood pressure, their temperature, you're taking some tests, maybe some ancillary notes have been written into the electronic health record system, et cetera. All that data that's pushed up to us and running on the analytics system within SAS, basically we're sending back to that practitioner this trend line of information. And so if it's a static trend line, that's great. But as the trend line goes up, that's when the patient needs to be looked at. And so based on the different algorithms that are in the data structure that we're putting together, that obviously scenario would protect that patient from hopefully spiraling down and having the practitioner see that patient much quicker. So for nurses, they would have the access to something like this, be able to, again, get an alert maybe on the system. They would then go see Jane Doe or John Doe to prevent them from going into a worse condition.
I think this is great. I used to work in the hospital system and I dealt with a lot of medical records, and just the pain of having to call Emory because I need this person's records from them and I couldn't access them to give to the therapist at Wellstar. It was very time consuming. And a lot of times you didn't even get the records.
Well, and here's a great question. So again, going back to everybody, raise your hand. How many of you have gone into a doctor's office and had to fill out the 5 or 6 page thing on the clipboard? Raise your hand. Yeah. Why? Because the data is not shared. So there you go. Yeah, big problem. And it's I think in the past, because everything has been done on paper, they didn't have the digital connection, but now that they do, the ability to transfer and open up that architecture of data is available. And so that's what we're working to try to make happen. Other questions? So let me do something real quick before I forget, because I wanted to get this before I got on. Do this. I'm going to text you guys something real quick or chat you guys something as soon as I get it back. Go.
I have a question.
Please.
So, your goal is to share data across multiple platforms on patient health, whereas to your doctor conglomerates or whatever, they use something like MyPrivia and share from office to office.
Correct. Yeah, basically it's taking the data, from the electronic health records. So any notes, nurse notes, doctor notes, any type of lab testing, anything that's a monitor, any type of prescription data, all of that will be wrapped in.
So my doctor's office was recently hacked. Their systems were completely hacked and shut down, and they couldn't make appointments for several months. I guess somebody wanted the medical data. So
yeah, that's
how
what
about the security of that. How does that work?
Yeah, well, that's totally understand. I had that with one of my doctors that happened too. So I feel your pain. So the way we're running everything, and really the aspect back to the research organizations, so the university research organizations like Emory as an example, we're working with what's called anonymized data. So data that doesn't have your name on it, but it has your information. So it's HIPAA compliant. That's the guidelines that the government has to make sure that your records are safe from a information standpoint. So we have that compliance through our Azure platform, which is a Microsoft platform, and GDPR, which is the European HIPAA. But we're working with anonymized data. So it's, if a hacking occurs, they would get information about someone's blood test or someone's blood pressure reading. They wouldn't know that it would actually be yours, Christy, individually. So that's kind of the uniqueness that we've set up. What happens is that there's companies that actually take the data out of an electronic health record system, and then they anonymize that data and they give it tokens. They give it basically a number. And so it's a cool thing, because if the patient of John Doe goes into the hospital, we'll know that it's the same John Doe that goes to the nursing home or is now treated at a hospital at home type of environment.
Thank you.
To opt out for security. Yes. Yeah, they basically can do what they want to with the information. If they want to opt in or opt out, they have the ability. One of the things we've structured the system on is the ability for research applications. So in essence, we give a tag to where if you want to be part and allow your information that's anonymized to go to research purposes, you have the ability to do that, or you can opt out. Other questions? Next question for you all. So raise hands. How many of you are chemistry teachers? Okay. How many are physics teachers? Keep your hands up if you could. Just want to see what the scope is here. You can keep your chemistry ones up too, if you can, if you don't mind. What about computer science or IT?
Maybe not too many of those.
No? Okay. And then I'm assuming mathematics? What am I?
Kind of stuck to
Biology?
science, but there might be
Biology,
some.
science? And then what ones am I missing?
We might have some healthcare teachers.
Okay, didn't know that was there. It wasn't there when I was in high school, but thank you.
Yeah, the CTAE pathway.
Very cool.
Environmental science.
Biology and forensics? You're kidding. Wow. Environmental science? Okay. See, these are all new to me. Okay.
You're learning something today too.
Yeah, that's very cool. And just for me, guys, and again, I mentioned the different schools that I went to. And again, you guys are teaching kids that are probably much more well educated than I was from an IT standpoint back in the day. But, I think giving them full reign and ability to learn, I think that's a huge driver. Obviously keeping them optimistic on, how they can better themselves and keep passion kind of as a thing in the forefront for what they want to do. Somebody asked earlier about nurses, and somebody who wants to get into that space. I mean, that's a tough role, and it's a tough occupation, but people love doing it when they get in. And so, to keep that passion for them, that's a huge win-win for the student and obviously the healthcare system. Within my setting, we've got everything from biologists to PhDs who work in microbiology to tech geeks. I've got a gentleman who recently retired from Intel. He did all of the CAD work for the Intel computers. So, I mean, we've got some pretty unique folks on my team. And again, they went through the high school settings like everybody else here on this call. And just, I think helping find their way, I think is a huge benefit to them and really to the future of all of our businesses within either healthcare or other. Okay, there we go. So we did that. What type of applicants does our company hire? I mentioned a couple. We did actually do some internships as well through UCF, and I should have left information on that. So we've got my IT and finance group is down in Jacksonville, Florida. And so through that, we've got some folks that we're connected into at University of North Florida, which is there in Jacksonville. But we also are well connected into USF, University of South Florida, Florida State, University of Florida still, and then UCF. And at UCF, they had students that were trained on SAS and the use of SAS, SAS, within their structured training environment. And so we had 3 students up until the summer of this year that were working on our platform. So that was a pretty cool thing. And we're looking to do that as well with them again. And then I'm also tapping into University of Georgia as well to do that. And anything else?
How soon would this technology be available to like the everyday ordinary person?
Q1, so January.
January, okay.
Yeah, so what's and one of the things that's really cool, you know, there's going to be imaging data. So one of the, I think the aspects that we're going to find is that if you've got an MRI or a CT scan that's done, let's say on a kidney or a heart, or you know, let's say a stomach issue, oncology issue in the stomach, you know, the scans that we'll be actually able to run AI on, artificial intelligence on, I think that's going to be the big breakthrough, because if you've got a patient who, and I'll go back to an oncology patient, a cancer patient, if you've got a cancer patient who's got a screened organ, and then you've got that same patient on certain medications, and then you've got that same patient on wearables or monitors, and to be able to trace and track that patient throughout a time period, that's going to be huge. And so there might be nuances of finding out that certain medications work better than others, that certain protocols work better than others, that certain tests run better than others. And so that's ultimately what we want to try to drive towards as well. It's just the ability to kind of enhance the current state of healthcare today.
And Eric, what would be the best way for them to follow your company through your LinkedIn?
Yeah, they can do that. They can go on the www.persone.com. There's the links up there. So there's Facebook, there's the Persone LinkedIn, and I believe there's a Persone Instagram, but don't quote me on that one. I know there's not a Persone TikTok. Sorry for all your kids. Can't do that. It's one of those.
Thank you.
Awesome.
Yeah, so, and guys, I'll post all of his links separately. I'll pull them out of the chat.
I'm going to put I'll put my, there you go. There's my email too. So if you have any direct questions, let me know. And if you ever need somebody to talk to your kids, would love to do that too. My hand's up for that. So I'll volunteer. I love doing that. It's always kind of fun, the questions you get. You might get more questions on the AI than you think. So there you go. Awesome.
Yeah. Awesome.
Well, Megan, thank you very much. Appreciate
Yeah, thank you, Eric.
Thanks for everybody's attention and questions, and good luck teaching those kids, okay? Appreciate it. I really do. Thank you.
Yeah, thank you. Have a great night.
You too. Bye-bye.
Awesome. So you guys can see that even if your students weren't considering a career in AI, they're, you know, it's showing up in places that you might not expect. So maybe they'll find some connection between these different things that they end up liking. AI is popping up in pretty much every company. So if that's the kind of thing that they're interested in, they could pursue it at a life science company, perhaps. So awesome. We will go over a couple more slides. Let's see. All right, back to my view. Okay, so just a little bit more about expectations for the next couple weeks and then in the spring. So hopefully everybody can participate in all the sessions. I know that things come up. I've had a couple people email me today that have had, you know, somebody has to go to the hospital or just they had an accident, something. If something pops up, just let me know and we'll make a mark. And then these recordings, I'll work to get them available by the Friday. So this one will be available this Friday. It'll be in the Google Drive in that there's the document that's called workshop recordings or something like that. So I'll post those there, and probably this Friday I'll email everybody to remind you of where that is. So please have your camera on if you can. I know sometimes toddlers pop in and out and you might need to turn it off, or students are coming over and throwing things on your desk since a lot of y'all are at school. So if you need to turn it off for a second, that's fine. And then just try to leave it on so we can see everybody. It's a lot more engaging and interactive, and it's more fun for the presenters to be able to see everybody. A couple things that we'll go over a little bit more detail later. So there will be an equipment scavenger hunt. One of the things we see is maybe you're teaching chemistry and you have no idea what biotech equipment is available at your school. So I'll give everybody a list with pictures so you can start going around your school and saying, hey, have you seen this in your stock room? So that way you know what's available already at your school for you to borrow. Excuse me, speaking of toddlers, I have a toddler cold from daycare, so you will be able to pick one lab either from these virtual trainings or one of the ones that we do at the in-person in the spring, and we will ship all of the supplies for you to do that lab with your students. So say you teach 3 sections of biology, you say, I want all of my biology to do it. We'll send you enough for all 3 sections to be able to do it so that you can just slide it right in. You don't have to worry about one section doing something different than another one. So you'll get the supplies, and then if you need to borrow equipment, we do have a loaning library, which I'll go into more detail about what's in there a little bit later. And our requirement to be able to borrow from that is participating in one of our trainings. So just making sure that I know that you guys know how to use the equipment. Let's see. And then the deadline for getting all that done will be mid-May. So that way we'll write our report and send it in to the Department of Education. We'll have some opportunities to share your implementation. So we'll have some sharing sessions, or if you miss one of those, you can just write a report on it. Just a quick, it's nothing extensive, but just a summary of what you've done, so that we can see how it went and how your kids liked it. And then we will have a program evaluation survey at the end that I'm not sure when I'll send that out. Honestly, I'm still figuring out what is everything that I need to do. So this is an adventure for all of us. All right, so a little bit more on the schedule. We have our 4 virtual sessions, and then the in-person will be at Central Georgia Technical College in Macon. I know originally the application said Warner Robins, but they were completely booked for our dates. So Macon is right up the road. It might be closer to some of you. Hopefully it's not too much farther for the rest of you. But the technical colleges have been phenomenal with helping us to host trainings. And then you guys get to see their beautiful campus. They have plenty of programs within life sciences and then outside of life sciences that maybe your students could be taking advantage of. And then the last thing here, we will have some focus groups available. Basically, that will not be with me, that would be with an intern, and then you can give feedback about the program in kind of a structured group setting. And then again, the report. So, and then our fall speakers, we just heard from Eric Doherty. So hopefully you guys, I think everybody found that pretty interesting, and you have something to share with your students now. Our next speakers, October 10th, will be Katrina Johansson from Next Gen Biobanking, and they do biorepository services. So that's a nice fun word that you guys will get to learn about. October 17th, we'll hear from Dominique Smith at Vero Biotech about their medical devices, and also Shalu Suri from the Cell Manufacturing Technologies group, which she's at Georgia Tech, but there are a couple different groups that they work with, including UGA, and then I think University of Madison, Wisconsin, or Wisconsin-Madison, yeah. And then the last one, October 24th, for the virtual series is Dave Penicky, and he works for Sale Therapeutics, and they do personalized medicine. So you can see that we're covering several different kind of areas that are available in life sciences and emerging areas that you may not be familiar with. So today we'll continue with our whole group. We're going to do the lab safety simulation and pipetting simulation together as a whole group, and then we will split you guys out into breakout groups. So does anybody have any questions right now that I can answer? Good to go?
I just asked one in the chat, but it's
I've lost my chat. Oh yeah, and I'll send out a save the date with a couple of recommended hotels. So yeah, sorry about that. Okay. Okay, let's see. We already spoke with Eric. Okay, so the safety simulation, we'll go ahead and start that as a group. And I'll pop that link in the chat. So this is a virtual option for lab safety. So if it's something that your students maybe need to do ahead of time and have a look at home, you can assign this as homework. Or you could have them, if they're using the computer lab, sit down and complete this together. And I'll actually bring up the, yeah, I'll pull it up myself. So normally we do this as pre-work, but we wanted to just take a minute. A couple of you did it as pre-work anyway, and thank you for that. But we can walk through it together. Well, not together, but we'll take a moment and you guys can walk through it, and I will have it open to look at if you have questions. But I'll share screen so you guys can see it. So the North Carolina Bio Network has several different lab simulations that you guys are welcome to walk through. They're all free, and I believe none of them are behind a wall, like they don't have to log in or anything to access them. So you could assign these through your class and not, you know, nobody needs to create any new logins. But you can see from the activity, it should give them an idea of the proper attire needed in lab space, define the PPE needed in the lab, so all that personal protective equipment, explain how to have a safe and productive lab environment, so keeping our areas clear, using safety equipment that's around the room, and then understanding why following directions is important. I don't think that can ever be overstated. So, and then, so following directions. Also, if you look at industry job openings, a lot of them say following standard operating protocols. So that is very much following directions. So it is a great skill to have and reading step by step what needs to be done. So if you're working in a lab, we need to make sure that no step is skipped. So I'll go ahead and launch the activity. So you can see there are some instructor resources down at the bottom. There's a PDF. So it'll bring it up, and then you just click anywhere in there to start. You can put your name in for your completion certificate. I see a couple getting emailed to me. And hopefully you guys have my email at this point, but it's in the chat if you need. So I'm going to enter my name. And then, so you can go through the introduction to lab safety activity. And then they have the different modules listed there. So I'm just clicking on the intro to lab safety. And then everything is in there. So they go through what is proper attire for a lab. So, and we know all of this. but you guys can click through on your own. And then you can launch the little activity about dressing up the person. So they tell them again what all they should be wearing to be properly dressed for the lab. So I have long hair. My hair should be tied. I should be wearing not a tank top. I'm going to put on long pants and let's have some sneakers. Great, I'm properly dressed. So some of that would not be dress code appropriate anyway, so should not be wearing it to school probably. Oh yeah, and if it's blocked for some reason at your school, please do it at home. That's crazy. I'll redress my person. And so then you have questions to answer as you go through. So what should she be doing about her jewelry, cell phone, chewing gum, and other personal belongings? My story for students was always with the bubble gum. We had like an anatomy lab, and so it's all the formalin that they're using for the preserved specimens, and that gets into the air, and then it gets caught in your chewing gum, and then your gum eventually will start to taste like the preservatives. And so reminding them that that's why we don't chew gum in the lab. That's part of it. Never mind the sticky factor. Oh, perfect, Christy, you got it going. Okay, and Savannah might be able to get to it. Perfect. So do you guys have any questions about this? Just working through it. Okay, I'll let you work in privacy. Peace. I see questions popping up in the chat, and I see some certificates coming in. So thanks everybody that's emailing them. I'll stop sharing so I can see the chat. Great. I'll give everybody a couple more minutes to wrap up. I'm seeing a bunch coming in. So if you're having trouble taking a screenshot, feel free to just put it on your phone and then email it to me when you have a chance. Just take the photo with your phone. Yeah, and one question. So when you get emails from me, they say at cghi.org, but at gabio.org also works. They go to the same place. So is everybody, is it pretty straightforward for everyone? So are you guys seeing where you could use this with your students? So are we thinking beginning of the year, or anybody want to come off of mute and chat? Obviously not mandatory.
This is a good one for
Vanita?
the beginning of the year. When we have to go over lab safety before we do any labs at all. I like it because it's interactive. It's not just a piece of paper that the kids have to look at. And they love to see things on the screen and click and whatnot. So I think it's a good one for the beginning.
Awesome. Yeah. And maybe a refresher after the first time that they are not following some of the instructions. Awesome. I'm glad this is useful for at least some of you guys.
I think at the beginning of the year, I can use this as well. They also have
definitely an upgrade.
I was going to say they also have a zombie college simulation, the same website, that's really good.
Yeah, so you guys definitely click around on there and see what else they have to offer.
Mm-hmm.
It's been a really good virtual resource.
It's probably because I'm on a Chromebook and I prefer a desktop, but I'm getting frustrated with the website because I don't know how to navigate back and forth if I need to go back. Like I don't see a certificate, so I don't understand like what people are sending a screenshot to you of. So I'm getting frustrated with the website personally. Okay, so it's not good for Chromebooks is what we've heard.
At least Christie's is not playing nicely with it. So sorry, it's frustrating. If you just shoot me an email and say, hey, I did it, it's fine.
But like,
what is it?
What's it supposed to like give me at the end?
So let me see if I can share what other people are sending me. I'm getting a lot of screen grabs of just this little, just it says, congratulations, you've successfully completed lab safety. There's Pam's. Yeah, so that's what shows up at the very end. Yep, and screenshots are fine. So if it doesn't end up giving you a certificate, just shoot me an email that says, hey, it didn't come up. Okay, and Amy said the students need to have the screen at 80%. So maybe that's some of the issue too. Yeah, and Faith, yours is good. Thank you.
I'm sorry, if you're on a Chromebook, maybe take your screen size and reduce it down. I was at 100 and I had to reduce it to 80.
Okay, is that just take your fingers and kind of pull it? I don't speak Chromebook.
Yeah, well, go up to the, what is that, the hamburger, the 3 dots in the top right-hand corner, and zoom down the whole screen.
Gotcha. Okay, so some Chromebooks are playing nicely, some need to be zoomed, some are. All right, and I'll stop share.
All right, if I'm having technical difficulties, if you ask me a question, I didn't hear it.
Oh, I don't think I did. You're
Okay.
good.
Okay.
Yeah, I've heard some echoing from some. It's just not feeding the speaker properly, I think. Awesome. Can I go ahead and show you guys the second simulation? Okay, and I'll pop that one in the chat. I did a tiny URL for this one. There we go. Just to make it a little more straightforward. So that'll work for you guys too if you want to just use that with your students. It's now a permanent resource on the internet. But I will share screen and walk you guys through a little bit of this. And this is a resource that you can use before you start pipetting. So I see Mark has already gotten out his pipetting practice Petri dish template and his pipette. He's setting everything out. Trisha's got out her colored water. Yeah, I see some love for Lab Exchange there, and they have tons of simulations. So if you're not using it, it's definitely worth checking it out. The good thing about this pipetting simulation, it has different levels. So when your students first click on it, they can select a level. So level 1, I've never seen a pipette before. Okay, level 2, I've seen one, I've maybe touched one, I might know what I'm doing with it. Okay, and then level 3 are your kids that say, oh, I don't need to do this because I'm an expert, like I pipette every day. Well, go ahead and complete level 3. Okay. So there's something for everybody. So you guys can also pick the level that you are comfortable with. Maybe let me know if you're a complete beginner in the chat, say beginner, or in what are you, level 1, 2, or 3? Yeah, Transformation Lab is awesome. We got some beginners, got some level 2 and 3. Yeah, so a couple of you guys have done some of our trainings before. So we've done some light pipetting. Awesome. Yeah, so this is a great, oh yeah, see, we used them in college and then haven't seen them since then. And some people, we've worked with some teachers who worked in biotech for years and said, you know, I used a pipette all day, every day for years on years. Jeff would probably agree with that. I know Mark and Trisha will agree with that. So where is this located? So this is on Lab Exchange. I'll pop the tiny URL in again so that you guys have it. So you just go to, if you type in tinyurl.com/labxchange pipetting, it will come up and link to their website. And I like that I'm sharing Chrome, so you guys can probably also see that I have literally 100 tabs open. So welcome to my life.
Megan, I can get to Lab Exchange, but I can't get to that tiny URL address.
Okay. I'll put the whole link in the chat. I think I'm over it.
So if I can get to it through Lab Exchange, I can access it, but I can't do it through that tiny URL.
Gotcha. The tiny URL is not playing nice.
No. Thank you.
You're welcome. Yeah, hopefully that works. Okay, so if you know that the tiny URL goes through your firewall at your school, then there's that one. But otherwise, there's a direct link. So I'll post both in the in the materials for you guys. So I'm going to click on level 1 while I'm sitting here. So some basics, which Mark and Trisha will review. So always make sure that you have a tip on your micropipette. I should have brought one home with me, but I did not. You have a nice boxed pipette from Mini PCR. It should be a 2 to 20 microliter pipette. I heard somebody's was not, which is a mistake on Mini PCR's end because I only ordered 20s. So let me know if that happened. Yours should say on the box 2 to 20 microliter. So micro is that little U with the tail at the beginning. It may just be a UL, depending on their type font. And then it will review how to use the pipette. So you'll feel when you start using your pipette that it has 2 kind of stops. There's one when you push down, you'll feel some resistance, and then you can push it past that. So the first stop is when you feel that first resistance, and then the second stop is all the way down. So they show you the reagents here, which are very similar to what you guys will be using today. You should have a couple of food coloring solutions. And then they have all the different sizes of micropipettes. You only have the P20. I think yours says H20, but same thing. And then the tip boxes for each of them. And here's the blotting paper and a trash can. So you guys can use one of your little beakers for your any tips that you've already used. Yeah, okay, so good question about the tips. So pipette tips, the 200 microliter fit anything below 200. So they fit 20 to 200 microliter pipettes, and they fit 2 to 20 microliter pipettes. Because they don't have a filter on them. If you get pipette tips with filters, they will be specific. So the 20s have the filter where it's low enough that the P20s will fit in it. And then the 200s would, it would be too far up for a 20. I see a question, is that Raquel? Feel free to shout out or type it in the chat.
Yes, you pronounced my name right.
Oh, yay.
Okay, so I know that you're recording this. When you finish, will we be able to go back and watch this again?
Yes, so I have to get it off of the internet and then put it on YouTube. So it'll take me until Friday, but it will be available for you to come back and access.
Okay. I just want to practice over. Okay.
Yes, yeah, so you can
I
rewind
get-
it.
Yes,
and
I get the
do it.
information first, and then I can go back by myself. That's why I'm staring so hard, because I get the information first, and it's like literally out here you playing over in my ear again, and then I can do it. That's all I wanted to know. Sorry.
Yeah, perfect. Yep, and I
Thank
will
you.
attempt to break the recording up, so into sections with our basic information, and then Eric's section, and then Mark and Trisha's cohort sections will be separate recordings.
Thank you so much. Thank you.
Yeah, you're welcome. All right, and instead of blotting paper in your kits, you guys have a pipetting practice card. So Mark, if you guys, Mark and Trisha have it, you can show them how that works. So here they have a little section on estimate the size of the blot. So if a 10 microliter dot is this size, they ask you to make your dot bigger until you think it's 20. So maybe that's 20. Okay, and I see Vanita's message. So just check that you entered it properly as cghi.org. It stood for Center for Global Health Innovation. So it could possibly be backwards, but that should work. I've been emailing from it all day, so hopefully. All right, so 20, maybe that's a little bigger. I don't know, I'll call it that. And then 15, maybe in between. So that's 10, that's 15, 7.5. So that's 10, so that's probably 7.5. And then oh yeah, little bitty dot. So let's see if it likes that.
I'm sorry. I may be the old geezer here, but I am completely lost.
Okay.
So I put 1 to 2. I think I'm actually a like -4. So
that's okay.
I'm on my little screen. I'm lost as to what I'm supposed to be doing at this point.
Yeah.
I
So
found
you
all
have
my equipment.
Awesome. Yeah, well, I think that's the important part. So maybe we can muddle through the simulation and then you can actually do the lab with Mark or Trisha.
Okay.
Were you able to get the simulation pulled up?
Mute. I have it pulled up, but every time I do anything, it tells me I have to have a tip before I do bleh.
Ah, yes. So
Let me
you're
come back
here.
in so I can see you. Okay.
Yes, okay, and let me move the chat so I can actually see. So if I want to, let's see, it says select the P20. So I have the P20, and now it says select the volume setting to set it to 20. And so let's see. This little, where to move it. Okay.
Okay, I think I got it now. I think I was missing directions on the side.
Yeah, so, okay. I got it now too. All right, so if you click on the 2 microliter where it has that in the little teardrop, you can change the size. And so it's saying, yeah, warning there, make sure there isn't a tip attached when you change the volume to avoid contamination. So that's a nice little hint. So your student should be changing the dial before they add the tip. Okay, so we changed it while it was on the rack, but in real life, you need to pick it up. So I'm going to save my volume. Okay. And so it gives you a note here. So about some basic care for pipettes, you never want to dial it past the range. So if the range is 2 to 20, you want to make sure that you're staying inside that range. So the highest it should go is 20 and the lowest it should go is 2. If you go outside of those, it will mess with the calibration. And then also they're not, they're not valid outside of that range. So you can't do 1.5 and expect it to actually be 1.5 microliters. So we save it and then we close. So that one is set. And then we want to use these tips. So it wants you to open the box first. So we open the box and then, yep. So we have our pipette. So it's just hanging in midair like it's in your hand. And so then we close the box back and it tells you to avoid contamination. So these are all, this kind of falls under aseptic technique in your classroom, but these are the steps if you're trying to keep things sterile to make sure you're doing it. So now we have the tip on our pipette. And so we've done everything over here. We have check, 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 check. So then we are going to click on the red die to open it and then take our micro pipette and make sure it's down in the solution. So it shouldn't be in there to, do they really do it that way? Okay. So you should push to the first stop and then put it in the solution. Usually you don't want to blow bubbles into your solution. But there's the first stop. Okay. So I probably went too far. But you can see you just push down on it and it goes first stop. And if you hold it down longer, it goes to second stop. So then I have red liquid in the pipette tip. So we're going to move it over and then go to the second stop to blot it. So first stop to draw it in and then second stop to push it out. Okay, and then so you can keep going, pipetting onto the different blotting paper. So it said, move it to circle A, dispense. So we did that. It may not have counted it because I didn't do it right. And then, so we want to put the tip in the trash. So it comes up with this handy eject button. So you guys will notice on the side, you have, I think it's a gray button. And so that one will push off the tip and you can eject it into the trash can. So then once you're done, you put it back on the rack. And I probably didn't do it in the order that they wanted, so it did not say check. But you can see that they can follow the rest of the directions. So in circle B, doing 15 microliters. I'm going to close my die. And then C, 7.5, and D, 2 microliters. So all of these measurements are going to take the 20 microliter pipette, just like what you guys are going to be doing today is only using a 20 microliter pipette. So do you guys have any questions about the simulation? You don't have to screenshot this one for me, but you're welcome to screenshot it for yourself if you need to remember where it is. Yeah, you're welcome. All right. So if you guys don't have any other questions, we're going to go ahead and break up into some breakout rooms. I should have figured out how to open those before now. I'll stop sharing. And then breakout rooms. We'll assign I'll let you choose your rooms. And so that should make it where we can also add you to those rooms. So I'm gonna first hit breakout rooms, and then I'm gonna share screen. So the overview of how we divided people, if you are middle school, you are going with Trisha. Yeah, so if you are in with somebody else, yeah, if you're in the same classroom, go into the same room, but I will share screen as to what my vision was, and then we'll see where you guys end up. So I'm gonna put Mark in group 1, Mark Peterson, and then I'm gonna put Trisha in group 2. Okay. All right, and then opening the rooms. And hopefully I did that right.
You did say Trisha, middle school will be with Trisha. I am high school, but you still want me with Trisha?
Yes, so if you're on the Trisha
Okay.
list here, which is the right, please go with Trisha.
Okay.
Yeah, if you guys want to switch, we can talk about it, but try this one first. Unless you guys are grouped together and I didn't group you together. And then please just go to one.
And how do I join a different classroom again? I'm sorry.
So in the control area, there should be a thing for breakout rooms.
What control thing are you talking about?
So in Zoom, I think if you hover at the bottom, I'm in screen share, so mine shows up a little differently right now. But there should be an area that has like 4 little tile looking things that says breakout rooms.
Now, I see it now. Thank you.
Okay, perfect.
Is Trisha room 2?
Yes. Yeah.
Thank you.
So Mark is room 1, Trisha is room 2. Great question.
Okay, sorry. I'm still a little bit lost.
That's okay.
I don't see those tiles that you were talking about.
Okay. And Adriana, you're going to Mark's room, so I think I
Yes,
can
I
go
know
ahead and-
that.
Yes.
Is
I'll go
is
ahead
it
and
behind
move
my
you.
chat? No.
It should be somewhere near chat.
In the chat. Okay, let me find it there.
Not in the chat, but you know the toolbar where you can see the chat icon? It should be next to or near the chat icon in that toolbar.
Okay, well, my chat is it's open right now, so how do I?
But if you
Okay,
close
now
it,
I made the
do that.
chat full screen. Does that help?
So do you know the little bar where you opened up the chat?
Okay. Yes, I see the chat now. Participants, this? No.
It should be near the participants. If you hover through that list, there should be
Okay,
breakout. But
I've seen, okay, hold on. Participants 8.
And Jeff,
Look
you can
for-
go ahead
on
and
the
go
far
to
right.
Trisha.
On the far right, it says more. More. Click on that.
More, yes, I see more. Okay.
Click on that, and then at the bottom it shows breakout rooms.
Okay, here. Okay.
Okay, thanks,
Thank you.
Jeff.
And I'm number 1?
Yes.
Okay.
Oh, and Ashley, sorry, it's being rude. Mark's room, so number 1. And Kimberly, number 1, which I can just assign you to it. Let's see. Jeff, you can go ahead and go to Trisha's room, so number 2. And then please record. Thanks. Vanita. So Vanita, you're going to Trisha's room. I'll go ahead and put you in there. And Wendy, you're going to Mark's room, so I'll go ahead and put you in there. Vanita, you're back. I'll put you in Trisha's room.